postdoctoral fellow with Dr. Mayo, and over the past 1 year, we've been exploring using next generation proteomics to understand sleep disorders and also circadian rhythm. And I thought I would give you a brief update on this new technology we've been using. And so, not to beat the dead horse again, but circadian time is regulated by suprachiasmatic nucleus in the brain, and it's influenced by cues from the environment, and the SCN modulates the melatonin production in the pineal gland. And measuring internal circadian time has many implications clinically, and yesterday we heard from Dr. Duffy that almost 17,000 genes measured, or half of them are circadian regulated, and among them 900 different genes were actually drug targets. So this is very beneficial to dose chemotherapy and also understand certain sleep disorders. So the main method to actually understand the circadian time is to assay dim light melatonin onset, and this is the current standard. But still, there is no proper way to actually look at peripheral clocks in the body. So the schematic on the right actually shows the three different circadian biomarkers, and here melatonin is the one which peaks in the night, and the cortisol is the one which peaks in the morning, and the core temperature is the one which goes down as we sleep. So bear with me. This slide actually indicates to us that different studies which have actually looked at trying to predict circadian time, and here we find that most of the studies use gene expression, which is not a perfect solution because it is often contaminated with white blood cells, which are very influenced by infection and exercise, etc. So to briefly summarize this, there were several studies which use gene expression, and most of these studies used gene expression, and the error rates in these studies were on not less than 2 hours. And so we think that a monocyte-based gene expression sample was used by Nicole Wittenbrick, which was called body time, and in this the prediction error was less than 1 hour. But the only problem was that the monocyte-based gene expression does not indicate the peripheral clocks and also the central clock indicated by DILMO. Is there a possibility to use other proteomic methods which can actually be sometimes more accurate? On the schematic here, we see that this is how the DILMO is measured, and this is in an individual with a phase delay disorder. You can see that the DILMO peaks later in the night, whereas in a normal person it does peak in the early hours in the night. So here I want to introduce to you an aptamer-based method in which to understand proteomics. And here historically proteomics methods used antibody-based methods or a mass spec-based method. But here an aptamer is a single-stranded DNA molecule which actually binds DNA which actually binds proteins. And these single-stranded DNA molecules are selected through a process called SALEX, where a diverse DNA and RNA library is actually repeatedly bound to proteins, and then the remaining strongly bound DNA molecules are eluted out. And this is performed many times to get a set of single-stranded DNA molecules or RNA molecules which bind very strongly to proteins. So the aptamer has its advantages because they often bind a native conformation of the protein, and they can be actually used in a high throughput setting, and they also have a high dynamic range. And lastly, they also have a very high affinity and a low binding value. This SOMA-scan assay was found by Larry Gold in Colorado, and he actually thought that if you would get many different aptamers together in a high throughput fashion, this would be advantageous. So the main thing here was we have to have aptamers with very slow dissociation rates, so that these are in the complex with proteins for a longer time to be assayed. So the process briefly works as the solute and the aptamer are in the complex are first incubated, and this the single-stranded DNA molecule has a photoclavable biotin tag, which is then cleaved off by binding to streptavidin, and this protein is then released, and based on that we can actually based on the amount of these single-stranded DNA molecules or RNA molecules, we can actually quantify the respective protein. So we in fact piloted many different panels of these SOMA-scan, and among them, the latest generation is one which has 5,500 different plasma proteins that can be assayed. Most of these belong to many different classes of receptors, transcription factors, hormones, and mostly the aptamers that bind these proteins are largely in the membrane-associated segment. To reassure that what we are measuring itself is are valid signals, we also looked at quantifying protein quantitative trait loci. This is an example from the CSF, and here in fact we can see that the quantity of protein levels is strongly regulated by single nucleotide polymorphisms all through the genome. In fact, we found that 13% of the 1,000-odd CSF proteins we measured had cis-acting variants. So this is the study design we followed to look at the circadian rhythm biomarkers. And in fact, here we profiled 9 individuals over 35 hours in a constant routine, and we took every second hour a blood sample was used to look at the plasma proteomic profile. And this was done by both the 1K and the 5K panel. These studies were conducted with Harvard with Dr. Duffy and Seisler. And here the participants had a 3-day entrainment program, and on the day 4 they started a constant routine over 35 hours. And this involved being awake in a fixed position under dim light. So we performed 2 complementary analyses. One is the circadian analysis, and another is the machine learning analysis. So here in the circadian analysis, we first looked at proteins which have a circadian waveform. Here we used autoregression to find periodicities, and then built harmonic regression fits for every protein to find the ones which have the best fits by AIC. And finally, we applied the false discovery rate to find proteins which have the best circadian fits. In the machine learning segment, we used Dilmore times, which are assayed in the subjects, and these were converted into Cartesian coordinates that is like time into the angle. And this data set was randomly split into 100 time points and validated on 53 different time points. And the SOMA-scan matrix of proteins, we only used a log normalization so that this model can be applied to a new set of samples. And finally, the model we used was a cross-validated tuned lasso with the L1 regularization. And we chose this because the number of proteins which we had was much larger than the number of individuals we had, and also to prevent overfitting. Looking at the circadian analysis, we did find that compared to the 1K panel, which I don't show here, but we found a 4-fold increase in the number of proteins that are circadian regulated. And among the 5,000 odd proteins, we found 6.8% of the proteins have a circadian waveform. Interestingly, we did find that pro-opium-melanocortin actually peaks in the morning, as indicated by, as it is a precursor to the production of cortisol. So with this more information, do you think we can get a better prediction here? So the 5K panel we used, we trained on 100 time points, and the response variable was an internal clock time from Dilmo assay. And here we found that the prediction accuracy was pretty good, within an hour accuracy, and the circular Pearson correlation was very high between the predicted and the true times. In fact, in 80% of the samples, we could predict the circadian time within an hour and a half, and 50% of samples almost within an hour. The only limitation in this site was we did not in this study was that we didn't all the samples belong to a single site, and in fact we could not validate yet on any other sleep disorder patients. I want to call to your attention these are the top proteins which are selected in at least 100 training instances of the model, and some of these proteins are familiar. They're circadian regulated, while others are new or novel. And we also found that many of these proteins, for example, are very tissue specific and could indicate peripheral clocks. For example, the proprotein convertase subtilisin is a very liver-specific enzyme, and while pancreatic hormone peaks in the pancreas, etc. So in fact, when we could actually find clusters of rhythmicity in the sense that after finding the phase of these circadian regulated proteins, we clustered the phases, and we found that we could find 12 different clusters, and all these clusters have a similar waveform. And within each cluster, if I would have to run a pathway analysis, I would find these pathways to be overexpressed. For example, we have protein processing and maturation in the cluster 5, and whereas hair follicular morphogenesis in cluster 3, extracellular matrix assembly in 6, so on. So this really gives us insight into what clusters are over-enriched in which pathway. So since our samples also had a constant routine protocol, we thought we would capitalize on this to find time awake since start of the study. There is a constant routine. So we modeled the time awake as a function of the SomaScan matrix of proteins and within each individual. So we did find that we could actually predict the sleep depth with high accuracy within 1.2 hours. But the only problem is that this could be confounded by the start of the experiment, and maybe we need forced desynchrony studies to understand this better. So in closing, we found that this SomaScan matrix assays actually indicate feasibility to study circadian regulation, and some of these proteins which are enriched actually represent peripheral clocks. So this could be used to study also the peripheral clocks in various tissues. This assay here using the 5K panel, we had some limitations such that there was only a single site, but still the predictor performed accurately. This remains to be seen if the predictor might actually perform well in real-world case scenarios. And finally, acute sleep debt could be measured, but the study was not designed here to pick up this accurately. I want to take this opportunity to thank Minyo Lab and also Dr. Ling and Zing from Minyo Lab for biobanking, and samples from Dr. Jean Tuffy and Dr. Seisler. Oops, sorry. Thank you.
Thank you very much. Questions?
Oh, I'm sorry, I have a very detail-oriented question. At one point, SomaLogic was free, people could use it very freely, then it was bought by China, and then only special collaborators were able to use the platform. Has that been opened up since, or is that still in a collaborative mode?
I think it's still limited to certain collaborators. That's my feeling. Yeah.
Hi. Hi, Aditya. Thank you for the great talk. Over here in the orange. Hello. Hi. Oh, perhaps you can answer this. I was wondering, what's the age range of the participants who provided the circadian samples? And I was wondering whether you had examined similar in kind of an older population.
I think the age, most of the participants were students, I believe.
I was a little bit puzzled. You threw in the last thing there about acute sleep deficit. Was that just because this is a sleep conference, or was there something in that data that was directly relevant?
See, here I was just indicating that it is possible to actually predict the acute sleep depth using
from that data.
From the constant routine protocol we had.
Okay.
So from a time since awake. But we are still trying to understand these results. So I did not present what are the top proteins which are associated with sleep depth.
Why did people have deficits? That wasn't part of it.
Sorry.
Part of the study.
No, they had a constant routine for 36 hours. Yeah.
So I have a question. Do you anticipate that a similar platform might be useful for giving us a measure of some breakdown in circadian amplitude? You know, in the setting, for example, you asked about aging, and certainly in neurodegenerative disorders, we're hoping to find better measures of some loss of circadian integrity. Do you see proteomics as having an avenue to that?
Certainly, I think it's so. Here, in fact, in this study, we did not have data from other designs. For example, inverted sleep, and other forced desynchrony, so on. But with the 1K panel, we in fact tried to predict the circadian time, and we could do it with high accuracy, actually. So I think this proteomic panel can pick up variations in circadian time, and also if person has a phase delay disorder as well. So compared to gene expression methods, where I feel that those methods all are contaminated by other cell populations, which may not be relevant to a circadian perspective. So I believe that the SomaScan proteomic platform could be something which can accurately predict circadian time in other real-world scenarios for it. That's what we are we believe it has potential, and we would like to actually pilot this into different scenarios. For example, long sleep, weekend sleep, and things like that. So, yeah.